everyone, welcome back for chapter 2. I'm going to break this particular chapter up into 3 lectures again, about 23 slides apiece. So let's just get going. This first lecture is going to talk about basic cell biology. So in this lecture we're going to be dealing with chromosome reproduction and kind of how chromosomes move around when new cells are being made. There are 2 ways that new cells are made, through mitosis or meiosis, all right? And during both processes, chromosomes have to go from the original parent cell to the 2 new baby cells, or daughter cells as we call them. So we're going to talk about the reproduction of new cells and what the general features of chromosomes are. So chromosomes are long strings of DNA in living cells that carry the genetic information or instructions for making the organism. And those sets of instructions are called genes. And chromosomes themselves are made out of deoxyribonucleic acid, or DNA. They're also made with proteins, histone proteins, scaffolding proteins, that keep DNA wrapped up and focused in one spot of the cell. And then it turns out that eukaryotic cells, which we'll get to in just a little bit, have DNA and proteins complexed together in a larger molecule that we call chromatin. Now, when I was an undergraduate, it drove me nuts. Chromosome, chromatin, DNA, which is it? All you need to remember is DNA plus packaging proteins, which can be called histones or scaffolding proteins, equal chromatin. Okay, so chromatin are all these things put together. So we are going to start with basic cell types. Basically, in living things, we have 2 different varieties of cells. Prokaryotic cells, which are the simplest, smallest cells, and that consists of bacterial cells, and another set of cells called the archaea. The archaea are interesting because archaea are the most ancient bacteria on the planet, and they were probably the bacteria that were around first. Eukaryotic cells are a little bit more complicated. Eukaryotic cells have a nucleus. All of them have a nucleus. And this includes single-celled organisms called protists, some fungi, so mushrooms, ringworm, things like that. Plants and animals are also eukaryotes. So for prokaryotes, I guess one of the things for you guys to remember, and if you look at the discussion, you'll be able to use this information in today's discussion. So prokaryotes don't have a nucleus. They have their DNA, which is actually, it looks like a donut. So you can think about bacterial DNA like a donut. And it's found in a prokaryotic cell in a region called the nucleoid. It is not a nucleus, it's the nucleoid. There are no membrane-bound organelles in prokaryotes, and what that means is that you don't have these enclosed spaces inside of the cell that have stuff inside. Instead, prokaryotic cells have a really tough outer rigid cell wall, which is made out of cellulose, it turns out, and it protects these cells from the environment outside. They also can contain other structures inside of that cell wall, like an outer membrane. There can be a peptidoglycan layer on the outside. There are a few other elements that you would learn about if you took microbiology. But prokaryotic cells also tend to have flagella. Not all of them, but some of them do. And so here's a picture showing you a real simple bacterial cell. It looks like a capsule. Here's the cell wall itself. I don't want to call it a capsule because the outer membrane can also be called the capsule. On the inside, you have lots of ribosomes. Ribosomes are made out of RNA. You might as well just kind of dedicate that to your memory right now. Ribosomes equal rRNA, ribosomal RNA. Right, here's the nucleoid, and they're showing you this spaghetti-like structure here, which is all the DNA within the cell. And this spaghetti-like structure is actually kind of like a tangled circular piece of twine or yarn that contains the genetic information to make this bacterial cell. This is the flagellum. Bacterial cells can have more than one flagella. The flagella helps the cell swim through a fluid environment. The plasma membrane can also be called the inner membrane, which is made out of phospholipids, just like an animal cell or a eukaryotic cell. The cell wall that I mentioned before is that cellulose layer here, real strong and rigid layer. And then there's an outer membrane, and this outer membrane can be modified in a number of different ways depending on the species of microorganism or bacteria that you're talking about. Now, eukaryotic cells, sorry, this is kind of moving real fast today. Eukaryotic cells are interesting because they do have a nucleus. So what that means is they have this cell membrane, this circular cell membrane within which DNA resides. That DNA nuclear envelope or nuclear membrane is the same, almost the same structure as the outer membrane. And interestingly, when we say membrane, a membrane is a membrane is a membrane. It's a phospholipid bilayer. So, in addition to having a nucleus that's surrounded by a membrane or a phospholipid bilayer, cells have, eukaryotic cells have an outer membrane. Some may have a cell wall, but the cell wall is different from prokaryotic cells. That cell wall, typically you're going to find in plants or in fungal cells. They may have a flagella and other structures. Eukaryotic cells have 2 or more linear chromosomes. So bacterial cells have the round donut chromosomes. Eukaryotes have sticks or linear chromosomes. And then some examples of membrane-bound organelles would be mitochondria and chloroplasts. Mitochondria occur in almost all eukaryotic cells, and mitochondria have their own little circular DNA. That's an interesting thing. Remember that for future chapters. Chloroplasts, which you'll find in plants. The chloroplasts are these green organelles that undergo photosynthesis to allow plants to make energy. Chloroplasts themselves also have their own little round donut-like DNA. Again, dedicate that to your memory. That's going to come back in a couple chapters. There are other membrane-bound organelles like lysosomes, endoplasmic reticulum, and the Golgi apparatus. And if you look at a eukaryotic or animal cell here, you'll see a listing of different organelles that are present in eukaryotic cells. So some examples would be, again, the cell membrane on the outside, the nuclear envelope or the nuclear membrane. The nucleolus right here, this region, I did want to bring that up because I want you to notice that it's a real dark staining area inside the nucleus. The nucleolus is not the nucleus of the nucleus. This is where ribosomal RNA is made. In other words, this is where ribosomes are synthesized off of the DNA strand, and we'll come to that later. Here's a Golgi body. Here are endoplasmic reticulum, the rough, the smooth. Here's the mitochondria. Centrioles, which are going to be really important for mitosis and meiosis. So, cytogenetics is a field of genetics that seeks to understand how chromosomes behave and what their composition are in a given organism. And so this allows people to look at, let's say, for example, the number of chromosomes or the actual structure of chromosomes and align that with potential distinguishing characteristics of an organism. So, for example, this would allow you to figure out a way to distinguish 2 really closely related organisms, 2 closely related species. It could be that one species has 42 chromosomes and the other one has 46. That is a distinguishing characteristic. So, when people are looking at cytogenetics, what you're typically going to do is look at either somatic cells. Somatic cells, soma actually means body, and somatic cells are the cells of the body. So that would be your skin, liver cells, brain cells, blood cells. Germ cells are different. Germ cells are going to be producing gametes. Gametes are egg and sperm, all right? And so these germ cells give rise to our gametes. And so these are definitely different from somatic cells. For the most part, your body is made up of somatic cells, and only one small group of cells is set aside to become germ cells during development. Unfortunately, we don't have time to talk about that in this class. So during cell division, when a cell has decided to divide in half to give rise to 2 cells, that is one way in which reproduction occurs in living species. So a living organism, a definition of being alive, is the ability to reproduce. And when we think of the ability to reproduce, we often think of reproducing offspring. But we also reproduce our cells on a consistent basis. So for example, we're always sloughing off our cells on the outer skin layer. Those cells have to be replaced. Otherwise, you wouldn't have skin left, right? So during cell division, we can actually see how the chromosomes replicate, and behave using a regular old light microscope. Each chromosome, each human chromosome, has its own unique shape, its unique size, and it has what we call a banding pattern, or these dark and light areas present along the length of the chromosome. And those are called euchromatin or heterochromatin. When we look at an alignment of chromosomes in an individual, we call that a karyotype. And karyotypes are very important in detecting the presence of genetic diseases in offspring. Oh, my apologies again. I'm not doing so well with this today. So, for example, if we were going to look at a karyotype of an individual, what we would do is we would take a blood sample and we would spin it down in a centrifuge. The supernatant, or the liquid portion, would be blood plasma, and the pellet would contain lots and lots of red and white blood cells. We'd get rid of the supernatant and then put the red blood cells in what we call a hypotonic solution. This solution would cause the cells to explode and release chromosomes into the liquid. After that, what somebody would do is, a technician, what they usually do is they have a certain type of a stain that they can use to dye individual chromosomes, and then you take a picture of the chromosomes, and using a computer you can clip out each individual chromosome. So you'd like clip this one out, and based on their size you align them and organize them as chromosome 1 from, who knows, mom, chromosome 1 from dad, chromosome 2, chromosome 2, chromosome 3, chromosome 3, and lucky for us, people originally who developed this technique started with identifying chromosome 1 as the longest chromosome and chromosome 22 is the shortest. Using the dye, you can see differences in staining patterns, especially in this chromosome 3 between the parents' contribution to the cell. The X and Y chromosomes are obviously different from one another. The Y chromosome is quite small and the X is quite large. But this picture here is what we call a human karyotype. Karyotypes can be done for all types of organisms, humans, dogs, cats, plants. You name it, you can do a karyotype for the most part. So in human eukaryotic chromosomes, actually not even just human, but eukaryotic in general, most eukaryotic species are what we call diploid. So you get half your chromosomes from mom, half from dad. So in humans, we have 46 total chromosomes, or 23 per set. So you have two sets, 23 from mom, 23 from dad. Dogs have 78. Fruit flies have 8 total chromosomes, or 4 per set. This is why fruit flies are often used for genetic studies. Not only do they breed really quickly and multiply very quickly, but they only have 8 chromosomes to analyze, which is, a lot less than the 46 that humans or the 78 that dogs have. So members of an individual pair of chromosomes, like chromosome 1 from mom and chromosome 1 from dad, are called homologous chromosomes, or a homologous pair. Homo for same, and logous for, like they match each other. And so these two chromosomes are usually nearly identical in size. There might be some mutation that's occurred. They usually have the same banding pattern. Again, there might be slight variation based on mutation or the ability to stain each one. The centromere, or that skinny little waist in the middle of a chromosome, is usually in the same location. And most importantly, each chromosome has the same set of genes, but not potentially the same alleles. So let's say, for example, just to really simplify things, you have the gene for hair color, blonde or brown. But the you have the gene for hair color, but the alleles would be blonde and brown. Sorry, I didn't mean to confuse you guys on that. And that's what we're going to start talking about in Chapter 3, are how alleles affect, or how they interact with one another to give rise to the ultimate phenotype of an organism. Now, the DNA sequences between the chromosomes are going to be almost identical. There should be about a 1% or less difference between homologous chromosomes. But again, those slight differences is what gives you variation. And the example here is the eye color gene, blue allele versus brown allele.